Good to be back, and I actually am going to violate the program here, the topic here, and talk to you about something that's related to neurovascular dysregulation, but I'd like to tell you about two randomized clinical trials that we're currently doing at Harvard partway through. So those two are as follows. One is the so-called LIFT trial, Life Improvement Trial. It's dedicated to both ME and long COVID. And the two drugs that we are studying actively are pyridostigmine, also known as mestinon, and low-dose naltrexone. And the second is an ongoing prospective study utilizing frozen muscle biopsies from the leg, and I'll tell you why we're doing that in a little bit. So first, the LIFT trial. These are the two drugs. I'm going to build a little case for using pyridostigmine. I'll say up front, Dr. Novak and his colleagues got us onto this. The dysautonomia folks, this is a super common drug used in POTS, and we began to find it useful in ME and then subsequently in long COVID. I'll build a case for that. I'll tell you a little bit about low-dose naltrexone, but I think I've learned that from Ollie and others in this audience, so I won't spend a lot of time on the proposed mechanisms of action. It's pleiotropic. There are probably a lot of good things that happen with it when it works. And then I'll present, in the spirit of LIFT, a preliminary trial that we've done. It's retrospective, tongue-in-cheek. We're calling it Baby LIFT, and I'll show you why it's relevant. And then finally, I'll get to the ongoing study. So pyridostigmine, why would we use this one? Wenzong produced a very nice paper, now in press, that where patients have told us, given us positive feedback for its use in both ME and subsequently in long COVID. This is how we think it works in these diseases, including POTS and ME/CFS and long COVID. So there's a critical juncture here, which Ollie knows all about, which is the sympathetic ganglion. He just alluded to it. And we have to remind ourselves that there is a cholinergic step here. So pyridostigmine retards the breakdown of acetylcholine in the neural synapse, and for myasthenia gravis, which is our FDA-approved indication for that, that's at the end plate of the motor neuron. But in these dysautonomia cases, and we think in ME, and this is quitting, the autonomic ganglion is a cholinergic step. There we go. Which we think is augmented by pyridostigmine. And then outflowing from this is a postganglionic fiber, which is adrenergic, and amongst other things, regulates blood vessel tone. Ollie just told you that he has evidence for vasodilation, and so do we, using invasive cardiopulmonary exercise testing on the venous side and perhaps on the arterial side as well. So this is how we think it works. And why do I think this? Part of it is that we did a study. So those of you who were here yesterday know that we do the invasive cardiopulmonary exercise test. So patients pedal away with a catheter in their neck and a catheter in their wrist and a mouthpiece and a metabolic cart. And we can get a lot of information about what ails them during acute incremental exercise. So we did this randomized clinical trial a couple of years back with the help of Philip Joseph, shown up here. It was 50 patients with ME, just pre-COVID. And they did their clinically indicated invasive CPET first. We identified the preload failure, which is the vasodilation on the venous side, and then they were randomized to receive a single dose of 60 milligrams of pyridostigmine or placebo. Tiny dose, one dose, kind of proof of concept here. If they got drug, their subsequent VO2 peak during a cycling exercise, about a second one, 45 minutes after the first, improved. And then the question is, how did it improve? It improved because cardiac output was higher if they got drug, actually lower if they did not, maybe the beginnings of PEM. And the mechanism by which cardiac output went up was improved filling pressures, here shown as the right atrial pressure, which got better. So this was improved autonomic function even with acute pyridostigmine. We think it's acting at the sympathetic ganglion, and these are some evidence, some data to suggest that's how it works. All right, so the second drug we're using in the LIFT trial and the baby LIFT trial that I'll show you in a second is low-dose naltrexone, I won't spend a lot of time on this, but briefly to review, it's a mu opioid receptor antagonist, and 10 times the doses that we use. It's used for alcohol and opioid use disorders. These are the doses we use. This is the one we're targeting in the LIFT study. I'll come back to that. We know from the work at the Griffith and other places that natural killer cell function is actually augmented. So it's a unique anti-inflammatory agent, unlike a lot of other rheumatologic drugs, and that it does not immunosuppress. It actually augments the immune system, the innate immune system. It works in large part, maybe not exclusively, by tamping down the toll-like receptor 4 pathway, which in turn downregulates NF-kappa B downstream and the production of pro-inflammatory cytokines. So it's anti-inflammatory. I'll also point out that nerves are particularly susceptible to inflammation. They lack the protective mechanisms through the trail pathway to protect themselves. So in a way, we may be treating some of the dysautonomia in ME and long COVID with this. There are some tantalizing clues, including some of the authors here, that in fibromyalgia it may work, and then in ME/CFS, by self-reported response of symptoms, it may work as well, and we've had a lot of luck with it in the clinic at the Brigham in Boston. So this generated a study we call BabyLift. This was a preliminary look at these two drugs used either alone or in combination, and it really paved the way to the prospective study that is ongoing. So BabyLift, we had a unique outcome, not so unique. It's called the SHAPE test. So these are not patients, but these are two of our exercise physiologists doing a 3-minute step test. And for those of you who haven't seen this, it's a metabolic card. It measures many of the things of the both invasive and non-invasive incremental cycling CPET, and gives us a read on aerobic capacity. But one very useful number that it gives us is heart rate recovery. So when the patient stops exercise, the faster the heart rate decays, the better, the more parasympathetic tone, the more aerobic health. So we've used that as a major outcome in this study using the two drugs that I mentioned. And here are some of the results. I'll hasten to add, in this preliminary look, we didn't have a lot of low-dose naltrexone monotherapy, only 11 patients, so beware, we may have a type 2 error here. But we have Mestinon alone in a pretty robust number here, and we show that at 1 year out, after using Mestinon, pyridostigmine, in general, 60 milligrams 3 times a day, as monotherapy, heart rate recovery is statistically improved. So I don't think the stats lie, and you can see the individual responses here. Over here is the combined treatment of Mestinon and LDN in 57 patients, and a very similar signal. We think not everybody, but most folks with heart rate recovery improving. And then there's this interesting group over here. They didn't get either drug. In general, they're healthier, so their heart rate recovery at baseline is better, and there's some stability, I guess, so that's somewhat reassuring for the study design. So this is BabyLift. This suggested to us, both anecdotally in the clinic and this particular retrospective study, that maybe these two drugs are useful alone or in combination. So LIFT, and this is the ongoing study, funded, and a special call out to Linda Tannenbaum from OMF for funding this large single center study. Another call out to Johanna Squires and Shreya Palawi over here, who have really put this on the map. It took about a year to get through all the regulatory hoops, and we've been actively enrolling over the last 9 months. So we're taking all comers clinically defined with both ME and long COVID who meet the Canadian consensus criteria for ME. So they have to have all the criteria there. They get informed consent. We've got 4 arms. Here's the placebo arm. Here's the low-dose naltrexone monotherapy arm. Here's the pyridostigmine monotherapy arm, and then there's the combination of the two. This is 3 months worth. This is the dosing regimen. I'm asked all the time, what do we use? None of this is set in stone. We've learned from others, and it seems to be a workable dose, but in general, we are targeting with an escalating dose over 4 weeks or so, Mestinon or pyridostigmine 60 milligrams 3 times a day, and or neither the low-dose naltrexone 4.5 milligrams a day. We have the ability to decrease if there are symptoms, and we've had to do that in one case. The outcome variables include a wearable device. This is the Garmin Vivo Smart. It's got a whole bunch of physiologic variables. They wear it throughout the 3 months. And in addition to that one, we got at the beginning and at the end, and really in one interval look, a bunch of omics with the help of many collaborators here, proteomics, metabolomics, transcriptomics, lipidomics, not shown, and peripheral blood mononuclear cells sent to Maureen Hansen. We've got Dr. Sommerfeld's FunCap here, a well-validated way to assess PEM and function, and then these other well-validated questionnaires that the patients get electronically. They download, and then with the help of Wenzong, we get this into REDCap. So here's where we are right now. I can't give you any results. We're all blinded. The patients are blinded. We're blinded. No results till the very end when we break the code. But this is when we started. We've got 25 patients enrolled, 5 completed, 10 additional patients who are actively being screened. We had 2 withdrawals, just a little comment there. Both of the patients who withdrew from with really kind of minor side effects got worse after they withdrew from the study. All right, that's the LIFT study. So that's ongoing, more in a year, I hope. This is the second study. So it's a muscle biopsy study that's designed to look at a phenomenon that we and the Amsterdam group have found to be very peculiar during exercise in ME and long COVID, and this is failure to normally extract oxygen at peak exercise out in the periphery. The differential diagnosis physiologically of this failure to extract oxygen and depress the mixed venous oxygen content and depress the VO2 is vascular abnormalities with shunting of blood away from the mitochondrion and intrinsic mitochondrial dysfunction, or both. And this study is designed to interrogate those pathways. This is a recent update of our invasive cardiopulmonary exercise testing results in ME and long COVID. We've got 450 patients with ME, another almost 90 with long COVID, and some normal controls. And I just show you this. This is the systemic oxygen extraction that's depressed in an equally prevalent way in both long COVID and ME when compared to normal controls. So this is a real contributor to the depressed VO2 peak and presumably symptoms, this peripheral phenomenon, inability to take up and utilize oxygen normally. So here's our hypothesis with this muscle biopsy study that I'm about to show you some results from, the baseline results anyway, and that is that in ME and long COVID there's poor O2 extraction in a subset of patients, and it might be related to intrinsic mitochondrial dysfunction. And additionally, in accord, I think, with the Amsterdam group's recent work, that this may worsen and contribute to post-exertional malaise. So here's the study design. We've targeted 40 patients with either ME or long COVID, so the ME had to be present before the onset of the pandemic. And then we've divided them into good extractors versus poor extractors. We're really comparing those two phenomena at peak exercise from a pre-existing invasive cardiopulmonary exercise test. We will add maybe more than this, but for now we're going to add 10 additional controls with fatigue but no PEM. So we see a lot of patients at the Brigham with heart failure and pulmonary hypertension and other forms of fatigue, but they don't have any post-exertional malaise. So we'll be comparing all these things, good extractors to poor extractors, and then to this disease control group. The protocol is that we will take muscle biopsy. Vastus lateralis is the site on the thigh. It's frozen immediately and sent to Baylor. And we do this on day 1 and day 8 of the study. And then on day 7 of the study, they undergo a non-invasive, prolonged cardiopulmonary exercise test designed to provoke a little PEM. Not awful, but some PEM. And then we've got a bunch of outcome variables, including the shape test I just mentioned. That's that 3-minute step test with a metabolic cart. and we've got lots of omics going every which way with all of our collaborators and peripheral blood mononuclear cells going to Cornell. We've completed 27 patients, so we're a little over halfway there. And what I'm going to show you next is some baseline data of the muscle biopsies. It's not the subsequent PEM resulting muscle biopsies. And we've combined them with 14 additional patients for a total of 41 from 2 prior muscle biopsy studies. So this is what we found, and I presented a little of this yesterday. It may potentially, if it holds up, be somewhat unique to ME and long COVID. And that what we find is an index of mitochondrial biomass, which is the citrate synthase function. It's a marker of the number of mitochondria or the biomass of mitochondria in the muscle. And it's decreased overall. Now, there's a range in fairness, but it really doesn't get up above 100%. These are normal compared to normal controls here. This is diametrically opposed to genetic forms of mitochondrial myopathies, where the opposite happens. So in those diseases, the primary problem is the machinery of the electron transport chain, and in a sense, there's a compensatory increase in the biomass of the mitochondrion. In this case, we have fewer mitochondria, and we don't know whether they're fused or not. Hopefully we'll learn more about that from morphology collaborating with Dr. Woest et al. In Amsterdam. So this is different, but presumably this is not a good thing to lose biomass of your mitochondria. The other thing that we have found, and this is preliminary because there are small numbers in the good oxygen extraction group, is that bad oxygen extraction, what I've alluded to is really the genesis for this study, is associated with a decrease in all the electron transport chain function. And this is normalized to the number of mitochondria or the citrate synthase. So bad oxygen extraction at peak exercise in the periphery is associated not only with a decrease in the biomass, but a decrease in the function of the electron transport chain, so shown here for complexes 1, 2, 3, and then 2 and 3 over here. If one is able to increase the electron transport chain function, it looks to be protective in terms of that O2 extraction, and presumably these patients have fewer symptoms and a better VO2 max. So this little cartoon, this is preliminary, but this is what we think, and I'm going to end with this. We think that in ME/CFS, and presumably in long COVID, though we need more patients in that category, there is some evidence of mitochondrial dysfunction that contributes to presumably symptoms. The hallmark in these diseases, as opposed to the genetic forms of myopathy, is a decrease in the biomass, the number or the biomass of the mitochondrion. And then you have 2 ways to go here in terms of energy production, the ETC, the electron transport chain function. And we don't know why you go each way. But if you additionally, to the decrease in biomass, have poor electron transport chain complex function, you end up with poor extraction of oxygen at peak exercise, presumably a depressed VO2 peak and worse symptoms. But if you're able, in a sense, in a compensatory fashion, to ramp up your electron transport chain function, it's somewhat protective, and you have less of a problem with systemic oxygen extraction. So that's what we've got so far, more to come. We hope, obviously, it leads to some precision medicine. Special thanks to several folks. This is Open Medicine Foundation. Again, a special call out to Linda and her group who's funded this. We've got all the folks at Mass General Brigham, including Peter Novak, who is here. And then we've got the Tompkins group and then the OMF group, many of whom are in the audience, and I thank all of them for their contributions and you for your attention.